All right. I'm now excited to kick it off to the Chief Science Officer of Viome, Momo Vujicic. He's presenting on No Longer a Secret, Food is a Medicine. And I'll kick it to Momo to further introduce himself.
Thank you, Kyle. Great. Hello, everyone. Thank you, Nora, for the great presentation. Thank you for pushing the field forward, and thanks to all the participants and other players in this revolution. So I'll tell you about our contribution to this revolution of bringing food to the forefront as medicine. And I'll be talking to you about Nutrition 2.0. And so my talk today will focus on biochemistry and math, because we're basically converting nutritional sciences into just biochemistry and math. There's no other pieces needed, but this talk is not going to be highly technical, so no worries about that. So I'll show you the punchline slide, which I will elaborate on towards the end. But basically, the punchline slide is that does personalized nutrition work that's purely based on biochemistry and mathematics? And we have here some very important data from 4 clinical trials that we performed. These are single-arm interventional trials where the only intervention is personalized nutrition, that's foods and supplements, for IBS symptoms, anxiety symptoms, depression symptoms, and signs of type 2 diabetes. And all 4 of them reached the statistical significance in terms of improvement of the symptoms. So we're very happy with this start of where we are. And let's now begin with the presentation. So we are trying to redefine nutrition. We think of nutrition currently as 1.0, which is it addresses nutrient deficiencies, and unfortunately it's used to promote fad diets that are the best for everyone, which is really incorrect. What we're working on every day is Nutrition 2.0, which is 100% data-driven. So it's just biochemistry and math. You obtain data, biochemical data, and you apply mathematical equations to it to generate or compute the personalized recommendations. So there's no involvement in humans, there's no anecdotes, there's no opinions, there's no subjectivity to it. It is highly personalized because each person's data are different. And then we are moving towards preventing and curing chronic diseases, cancers, and aging using this objective Nutrition 2.0 approach. So what does this look like that we're building? What we think of as a future of healthcare for chronic diseases, cancers, and aging is basically a self-care. The technology will enable everyone to take care of their own health, so they won't need to wait until they're sick and go to the hospitals. They basically maintain their health, and it'll be fully preventative, fully personalized in all of these categories, and it'll be delivered via an app. And so the app will tell people exactly what to do, when to do it, what not to do, and it'll give them risk factors for essentially every chronic disease and cancer. We're starting to work on this dashboard. The most important thing about this, I think, is that it's going to be free to everyone. If everyone is asking, well, how much does this cost? It's eventually, and we're thinking 2 to 5 years from now, it's going to be free to everyone. And the reason for this is once we show that this is actually preventative and we have good health economics data, whoever is paying for the healthcare of people today, they're going to want this instead of paying for healthcare because the return on investment will be huge. So this future is what we are building. So I'm not speculating on that future. I'm not the one to sit and read about and wonder what's going to happen. Our company is actually building this future, and we are following this motto every single day. And so what does that look like a little bit sort of focused on our company? So our company is a health platform, and that's in this box here. And health platform includes many technological pieces such as clinical grade molecular data from stool, blood, saliva, any other human tissue. It includes clinically validated metadata, clinical grade bioinformatics, translational science, artificial intelligence and machine learning, and then a digital clinical research study platform that can bring enormous amounts of data globally. So that's the health platform. The best part about this technology is that it's open to the whole world. So just like the App Store is open to the whole world, our health app platform is open to the whole world, so that what we've built over the last 11 years and spent $200 million is available to the whole world. We're not keeping it just to ourselves. The potential of this platform is unlimited. We're currently in this particular space of food as medicine, focused on these two applications, which is nutritional therapies and precision supplements. The rest of my company is also focused currently on many other applications in the health space. They're listed here. And we're starting to already plan and start to execute on our longer term applications to eventually, 5 to 10 years from now, include planetary health. That means every single ecosystem that you can imagine on planet Earth will be supported by this health platform. Just a little bit about my company. So Viome Life Sciences is 5 years old now. We have now 170 employees. We serve over 300,000 customers in 93 countries. I'm very proud of that for a 5-year-old company that has not only survived, but really thrived. And to me as a scientist, this number is really something that I'm proud of. We have so far sequenced or generating, these are raw data from customers and study participants. We have sequenced 450 trillion RNA bases. And this is far, far bigger number than, for example, the amount of data that 23andMe has gathered. And we're now obviously hitting the stride and we're accelerating even faster. So this is really what powers our entire platform. And we only have one mission, and that is to prevent and reverse chronic diseases, cancers, and aging using all of these resources we have. So let's talk about our platform a little bit and what is systems biology and the power of RNA. I really would like one of the takeaway points for the audience from today is to understand that really DNA does not have the power to tell us about chronic diseases and cancers and aging. It's really the power is in RNA. So it's a far more informative and useful molecule. So let's go over why that is. So first of all, systems biology. We have been looking at the human genes for the last 20 years, and we just realized recently that only 5 to 10% of the contribution to the chronic disease and cancers comes from genetics, that it's really environmental factors, and that the microbiome plays a vastly larger role, a dominant role in our health. And that's really shown here with the numbers. Human genes are limited to 20,000, whereas microbial genes on an average person number about 10 million. And of those 10 million, about 1 to 2 million are expressed. So we are basically mostly a microbial world and influenced by that microbial world a lot more than our own genes. So in order to understand chronic diseases, cancers, and aging, we absolutely have to study the microbial genes. And we study them at Viome, mostly in saliva and stool, but we also study them in other sample types. And here is a couple of slides on the power of RNA. So you guys are probably familiar that many chronic diseases go through stages of relapse and remission. And so here's a classical one, which is IBD, inflammatory bowel disease. If you guys don't know, inflammatory bowel disease sufferers go through periods of remission, which is a healthy colon. They can eat anything they want. They can have a normal life. They can have a social life. And then just a few weeks later, their life turns upside down. Their colon becomes inflamed, and they're no longer able to eat anything. They cannot have social events because they have 30 diarrheas per day. They're in pain 24 hours a day, and their life is absolutely miserable 24 hours a day for sometimes months. What is the difference in DNA content? This is all human tissue. What is the difference in the DNA content of these two? Nothing. It's the exact same DNA. The DNA doesn't change in these two phases. So if you analyze DNA any number of times, you're going to get the exact same result. This DNA is telling me that this person could be healthy or could be very sick, and I don't know. That's because DNA does not actually hold that information. It's the RNA. And that's already been very elegantly shown that the gene expression profile of the healthy tissue versus inflamed tissue is very, very different. So if you don't measure RNA, you can't distinguish health from sickness. When you step down to the microbial world, the same rules apply. Here's an example I'd like to give where Clostridium difficile, one of the worst pathogens that is present in the United States, it kills a lot of people in the United States. That same pathogen can actually be a commensal, very healthy bacterium producing butyrate for us. So it's living there as a friendly bacterium. Only when we disturb that piece by introducing antibiotics can that same bacterium turn into a very powerful pathogen and kill the person. And so presence of the bacterium as measured by DNA can mean either health or death. And it can't distinguish, DNA analysis cannot distinguish those two states because it's the same exact bacterium. So DNA presence doesn't mean infection. But at the RNA level, when this bacterium changes from a friend to a foe, the gene expression profile massively changes, and it's very easy to distinguish. And this is why at Viome, we don't sequence DNA, we only sequence RNA. And it's been a very powerful tool for us to understand both microbial and human gene expression and their connections to diseases. And so, in addition to the power of RNA in terms of distinguishing different behaviors of bacteria, we can also distinguish which specific genes are expressed and to what level. And so in any kind of an ecosystem, you have a relatively small portion of the genes are expressed, and their expression level changes very wildly depending on the environment that they're in, whether it's a healthy state or a disease state. And those are the things that we actually measure quantitatively with our tests. So how can we use this information to harness the power of the microbiome? I'm giving you here an example where the same microorganism called Faecalibacterium prausnitzii, which is considered to be a healthy microorganism, can either produce butyrate, which is healthy to us, or not produce butyrate, depending on which diet you feed it. And so you can actually modulate whether a bacterium is going to be good for you or bad for you by two different diets by providing it with specific ingredients. All right, so let's talk about our direct-to-consumer personalized nutrition. So what does that look like? Any person can submit their samples. These are stool and blood and soon saliva samples as well. And what we do is we measure the biochemistry. That means we do RNA sequencing, and we identify all of the microorganisms that are present there and all of their biochemical activities. That's the biochemistry. We then overlay on top of those biochemical data mathematical algorithms that we learn from machine learning, and we produce personalized nutrition and lifestyle recommendations. So it's that simple. Samples go into biochemistry, math algorithms are used to compute the personalized nutrition, and that's what the person is delivered. This is a little bit now detail about our current direct-to-consumer tests. These two have been available now for quite a while, and saliva is being productized right now. We have very small sample collection tubes. Even blood is collected at home from a finger prick. These two tests are clinically validated. They're clinically licensed in the United States, and we have peer-reviewed publications for both of these tests. For saliva, we are working currently on the clinical validation, and the peer-reviewed publication has been submitted for review. So it's under review, and I will show you those at the end. Let's now talk about our machine learning. So these tests generate molecular data. How do we convert those into personalized recommendations? We do very large clinical studies in order to do that. So we have enrolled now almost 100,000 clinical research participants in our studies in order to derive very useful data from that large of a population. So I'm just going to briefly mention here, this is also available as a preprint. It's under review. I'm going to briefly discuss this project where glycemic index, which is how high your blood sugar is going to spike depending on what you eat. The glycemic index that you find by Googling the internet is basically for the average human. And likely none of us here are the average. We all have different responses to different foods. And that's very nicely shown here in our paper, where if you feed two different people the exact same banana, you get one person to spike sugar very highly and the other one to stay low. And the same thing with sprouted grain bread. You can see this person has the opposite blood sugar response to this one. So this is called postprandial glycemic response or glucose response, PPGR. And you can see how personalized it is, or it can be. So what we've done is we enrolled 1,100 participants in the United States and Japan. We collected either just their stool or stool, blood, and saliva at the start of the test. So half of them collected just stool and half of them collected this full set of samples. And then for the next 14 days after the sample collection, they consumed a carefully designed diet, which is detailed here, 104 different study meals with different composition of carbohydrates and proteins and fats and so on. They consumed a total of 40,000 meals that we actually provided them. So we have a very large account with Whole Foods, and 20,000 meals of their choice for a total of 60,000 meals. And now we applied machine learning to this massive data set where we told the machines, because these people were on continuous glucose monitor for 14 days while they were consuming this meal, the machines actually have the ground truth in terms of what is the glucose response to each one of these meals. but they also have the molecular data that are provided to the machines. And so the question we're asking the machines, given these meals and given this glucose response and given these molecular data, could you actually have predicted the glucose response for each meal, for each person, without actually measuring it, simply by doing this test? And the answer is yes, we can do that very accurately, in fact. So with 80% accuracy, we can determine the glucose response for every kind of a meal and every person. And so this is the machine learning where we did both machine learning and then validated that on a very large data set of independent test data. And so you can see here that has been converted now to our application where each customer of Viome benefits from this. And for any food that would cause high sugar spikes, they would be given an avoid recommendation saying, hey, you should avoid that food and you should try substituting with a different food. And so for someone, maybe couscous, for someone, maybe something else. Sometimes even almonds will show up as producing a high glycemic load, even though people don't think of almonds as having a high glycemic load. So this varies from person to person. So just one more slide on machine learning. I just want to show you the power of our platform and how large it is and how it's going to expand from now on. When we attacked prediabetes and diabetes, you can see here the size of our study. We actually enrolled 56,000 participants in our study, where we had 50,000 non-diabetics, about 462 pre-diabetics, and 229, so 51,000 total, participants. And this is the breakdown. And based on these numbers, we were able to develop machine learned algorithms that can basically distinguish healthy people from pre-diabetics and diabetics. Even these two are different from each other. And so now we have basically a molecular way of measuring how close someone is to prediabetes and diabetes, or how close someone is to being healthy and not having these two conditions. And this risk score is what we use as an endpoint for our clinical trial that I will show you the result of in just a couple of minutes. So how is this all of machine learning and computation and this biochemistry and math projected? It's simply delivered via our app. So every single food and every single supplement is listed. For all the foods, they're listed into 4 categories. These are superfoods, meaning eat every day, a lot of them. Enjoy foods, meaning eat very, very frequently. Minimize foods, try to avoid them if you can, but it's okay to eat some. And then avoid foods, meaning never eat them. And so these are more than 400 foods that are loaded into the app, and every person gets a different recommendation. So there is no such thing. Our computers are not aware of ketogenic diet or Mediterranean diet. Every single person gets calculated their diet. And then we also display in the app supplements, exactly which ones the person needs and how much of it they need. But we also offer those supplements as personalized for every person. So if someone orders a supplement pack, it will actually be made just for them and shipped to their home so that every person gets exactly what they need and nothing that they don't. So this is sort of a new area that we've had to enter because none of the current supplement companies wanted to do this because this reduces their revenue. We don't actually just sell as many supplements as we want to. We only sell the supplements that the person needs. So let's talk now about the efficacy of this data-driven personalized nutrition. So if you remember, the risk score that we calculate tells us how close a person is to being healthy versus being pre-diabetic or diabetic. And here we performed a large prospective study. This is a clinical trial that's blinded to the participants, but it's a single-arm trial, so it's just an interventional trial, and we enrolled about 3,000 people total. So half of them self-reported as low adherence to the recommendations, and half of them responded as having very high adherence to our recommendations. And you can see here that the risk score went from 0, where they started, we dropped it by about 20% for the low adherence people, and the high adherence people dropped it down by about 40%. And so these are very significant numbers and very, very promising for now for us to keep moving in this direction to show that our personalized diet actually can not only lower HbA1c, but also prevent diabetes. And so we are now obviously going to move into randomized, blinded controlled trials so that we can hopefully show that this works in a randomized controlled trial fashion. We applied similar strategy to many other conditions, and the ones that we prioritized are IBS, depression, and anxiety. And you can see here the results for IBS. This is the severe group of IBS patients, moderate and low disease activity in blue before the start of the intervention, and then in orange, the scores after the intervention. And so time point 1 is blue, time point 2 is orange. And you can see here that p-values for the reduction in all 3 cases are very significant. And you can see here the summary of the reduction of the IBS SSS scale for their disease severity score. The mean follow-up here was 160 days. So we're very, very proud of these preliminary results. But I do want to say that these are single arm, even though these are blinded trials, the people did not know that they were in an IBS trial. We really need to run randomized controlled trials, and those are going to come up soon. With depression, I'm going to just show you the numbers here. They're significantly lower than at the baseline, significant reduction in the PHQ-9 scores, and PHQ-9 is the clinically validated survey for depression. For anxiety, very similar story. You can see here the score reduction. And then we are applying the same approach, data-driven, machine-learned algorithms for about 30 plus chronic diseases. So I'll just flash a couple of these for you. We're very interested in aging. So we have built state-of-the-art aging models, both based on blood and stool. And this is actually now accepted for publications in Nature Genomic Medicine, but preprint is available for viewing now. So you can read about these. And what's very highly of interest to us is not just to tell you the biological age, which every Viome customer benefits from that, but to actually understand how to modulate the gut microbiome and the human gene expression profile in order to actually reduce and slow down aging. That's really our holy grail. And the same thing here for IBD. We have a very, very advanced machine learned model on a fairly large group of study participants, where we have 83% accuracy of distinguishing healthy people from people with IBD using our machine learned algorithms. Here, as promised, are our publications. We have 4 peer-reviewed publications. We have 4 publications that are available as preprints, but they are under review. They've all been submitted and are under review. And I'd like to thank the organizers. I'd like to, again, thank every one of you who's pushing this field forward. This is extremely needed. And we are a US-based company. We're mostly in Seattle. Most of our employees are in 2 locations in bright and sunny Seattle. But we do have quite a lot of data science and bioinformatics and systems biology in New York City, actually right around here in the Fashion District. So I will take any questions if there's time left.
Incredible presentation, Momo. Thank you for taking the approach that you have taken, so research-backed, showing us all that research that you guys have published. This is extremely fascinating and really going to help propel the field of precision nutrition. Extremely, extremely exciting. Thanks so much.
Of course, we enjoy it every day. We really need to bring biochemistry and math. Those are very objective sciences. It looks like we have a question. Thank you.
So it looks like this question is, most likely the consumers who are subscribing to your services are consumers that are very much health aware and have resources to pay for your solutions. How can you make your solution available to nutrition insecure people who may benefit the most?
Yeah, so absolutely. So we are, obviously, that's in our vision. We want to make this available to all 8 billion people on the planet from birth to death. That is, we are not going to stop until that is achieved. The really key step towards that goal is when we show health economic data and efficacy data combined, such that someone like the NHS in the UK and self-insured large corporations say, hey, this has a huge ROI and we're going to invest in it, and we're going to actually pay for our members to have access to this so that every member can get the tests and the supplements delivered to their home and they can prevent these diseases. But we still have to perform quite a few randomized controlled trials, and we're already working on those. So I would say sometime in the next 2 years, I think that the dominoes will start to fall, but it may take a couple of years extra. We were hoping to be sooner, but COVID really slowed us down by a year and a half. So in 2 to 5 years, I think the dominoes will start to fall on that.
Okay, great. If those are all the questions, we're going to wrap this up, and we will be moving to our next session very shortly. Thank you, everyone.
Thank you, everyone.